Okay, so today I want to give you an overview of the work that we are doing in our laboratory, which is focused on understanding the role of mitochondria, the function and dysfunction in cancer. I have a disclaimer to do. I am not going to present any direct metabolomics, but I'm going to show you some data on metabolomics and, more importantly, how we can explore metabolomics to understand molecular mechanisms of disease, including cancer. Well, you might be asking why our laboratory is interested in mitochondria. Maybe you might wonder what these organelles are. And I wanted to introduce you to the mitochondria. So this is a cancer cell. Okay, maybe, where should I move? This one. Okay, so this is a cancer cell. And we stained it with two dyes. One stains the nucleus, which is now in blue. And the other organelles that we stain in this image are the mitochondria. And you can see the mitochondria are these beautiful red structures. Now they are red because we colored them in red. But you can see these are spread all out the cells. And why they are important? You might know that mitochondria are essential organelles for the life of the cells because they produce the majority of cellular ATP. But we now know that mitochondria play main functions in cells physiology, from calcium signaling to program cell death, and of course for us, metabolism. And you might have heard about the work of Warburg. Warburg was a pioneer in cancer metabolism more than 100 years ago. He demonstrated that these organelles, the dysfunction of the organelles, is actually the driving source of cancer. However, although more than 100 years have passed from the work of Warburg, we still don't know whether there is a comprehensive role of function, the mitochondria function and dysfunction in cancer. So we recently capitalized on the large collection of data from the Cancer Genome Atlas. The Cancer Genome Atlas encompasses, now we have more than 20 tumor types, more than 8,000 patients, and now I think probably we reached 10,000 patients. And we have data from all these tumors, RNA gene expression. And we can use this gene expression data to understand whether metabolic enzymes, including mitochondrial enzymes, are dysregulated in cancer compared to the normal tissue. And what you can see here basically is this pipeline, and our goal was to identify metabolic features that are present in the tumor compared to the normal for each tumor type. But we didn't stop here. What we did was to compare the gene expression from the low survival patient to the high survival patient from each individual tumor types. And the idea was to identify metabolic pathways that would correlate with the clinical outcome of patients. So when we analyzed this data, we were quite surprised to notice that the most significantly enriched and suppressed gene signature across all the tumors in the poor clinical outcome patient was this signature called OXPHOS. And I'm not sure whether you know what this acronym stands for. It stands for oxidative phosphorylation. And this is the key pathway in the mitochondria that regulates ATP generation. So this result suggested this view that I mentioned at the beginning, that at the origin of cancer, at least in this cancer patient with a poor clinical outcome, there could be a suppression of oxidative phosphorylation, therefore a suppression of mitochondrial function. Of course, this data supports the general idea about the role of mitochondrial dysfunction in cancer, but we didn't know why these patients were having this poor clinical outcome. So what we did was we probed the same cohort of patients for what is called the hallmark of cancer. So these are 50 and more gene signatures, or oncogenic gene signatures, for which we know how they cause cancer and how they actually exacerbate cancer progression. And this is just an example of what we found, which I think is quite remarkable. We found that in the patients where OXPHOS is suppressed, so where mitochondrial function is low, there is a parallel increase in many oncogenic signatures. But the most important for us is this EMT, which stands for epithelial to mesenchymal transition. And I'm not sure whether this is ringing any bell to you, but I'm presenting very briefly what the concept is. Epithelial to mesenchymal transition is a process whereby the cell of origin of tumors that we investigate, carcinoma, that are epithelial cells, indicated here, undergo this profound epigenetic and phenotypic rewiring where the cells become mesenchymal, and this means that the cells become more motile, more invasive, and this primes the cells for metastasis.
Okay, so this is why EMT is known to contribute to cancer initiation and progression, because it's linked to metastasis, which, as you know, is the primary cause of cancer death in patients. So this data seem to suggest a slightly different paradigm now. In our cohort of samples, we found that OXPHOS suppression is correlated to this poor clinical outcome of patient, and probably the EMT is what is driving this poor clinical outcome, likely because of metastasis. Of course, this is very broad, and we cannot infer actually any of this information from the Cancer Genome Atlas or the dataset that we use, because we don't have information about metastasis. So what we did was to use another cohort of samples. This is a melanoma dataset, where we actually have information about the gene expression from different metastases of the patient compared to the primary tumor. So here we can actually compare the gene expression of mitochondrial enzyme in metastasis, different metastases in the same patient compared to the primary tumor. And what we found is indicated here, very strikingly, we found that OXPHOS, again, the suppression of OXPHOS is a key feature of metastasis. So this corroborated a little bit, again, the link between OXPHOS suppression, the poor clinical outcome of patients, and the EMT that can correlate with metastasis. Although I'm sure that you are definitely not happy about this, this data that I'm showing, because we cannot infer causality from this data set. We cannot say whether OXPHOS suppression is what drives cancer and what is driving actually EMT, or vice versa. Actually, it can be that metastasis is what is driving this OXPHOS suppression. So in the lab, we are currently working on many different models where we can investigate this, and we can try to disentangle this connection. And in particular, one model that I'm going to show you is a model where we were able to understand the process of metastasis and try to understand how mitochondria contribute to this. Okay, so this model was generated by Zachary Van der Ranta when he was a postdoc in the laboratory of Joan Massaguet. What he did was very clever. He generated some metastatic cell line from clear cell carcinoma cell line. This is a cell line that is quite established. This is code 7860. What he did was to inject the cell line in the tail vein of the animals, or the mice in this case, and to harvest the metastases that emerge in the lungs. However, to generate a proper metastatic cell line, he had to repeat this assay many times, and he obtained this cell line called 786M1A, which is a metastatic variant of the primary cancer cell line. And if you inject this cell line into the lungs, into the tail vein of the animals, the metastasis burden is much higher. So these cells are two isogenic cell lines, same nuclear background, they differ only on the basis of their metastatic potential. Okay, so here we can now understand whether metabolism plays a role in this transition, in progression, and in particular whether mitochondrial metabolism plays a role here. So we performed one of our favorite type of assay, which is metabolomics, based on liquid chromatography coupled to mass spectrometry. And we were really surprised about this data, because these cell lines that are very similar, they have the same growth rate, very identical properties apart from the metastasis, have a very different metabolic phenotype. They can be distinguished by their metabolic signature. And we are currently trying to address this more in detail, but I'm not going to show you much data about this. I just want to confirm that in this metastatic cell line compared to the parental, we can see a striking suppression of oxidative phosphorylation, and we can also see a striking suppression of oxygen consumption rate. This is an acronym that stands for oxygen consumption rate. We could measure it in normal epithelial kidney cells, HK2, and you can see that this oxygen consumption rate goes down the more we progress towards metastasis. So this seems to corroborate a very important role of mitochondrial dysfunction during the progression. But again, I'm sure that you are not satisfied, because here we are still looking at correlation. But here we have an example where a genuine mitochondrial dysfunction can cause cancer. And this is a model that I'm going to describe very briefly. This is loss of fumarate hydratase. That is the cause of a very specific form of renal cancer. What? Okay. Yes, so fumarate hydratase is the enzyme that I'm going to describe very briefly. This work that I'm going to present has been carried out by Christina Schmidt, a PhD student in the lab, and Marco Sciacovelli. So just for some of you who might not be familiar with fumarate hydratase and with the Krebs cycle, so this is the mitochondrial before, now it's green, and this is the Krebs cycle. And the Krebs cycle, as you know, is a very important pathway in the mitochondria because it converts nutrients to reducing equivalents for the generation of ATP. Fumarate hydratase converts fumarate to malate, and this reaction is very important for the Krebs cycle, but genetically is very important because in 2002 it was demonstrated that fumarate hydratase mutations drive this hereditary cancer syndrome called hereditary leiomyomatosis and renal cell cancer. This is characterized by benign fibroids of the skin and of the uterus and an aggressive form of renal cancer, which is called papillary type 2, and it's highly aggressive and metastatic. So this was a mystery. When this came out, nobody really could figure out why a mitochondrial enzyme, if mutated, would drive cancer. And it's still unknown, but or at least partially known, as I will show you. But one of the biochemical features of fumarate hydratase loss is the accumulation of fumarate, which goes to the high levels in cell lines that we generated, but we could also find high levels of fumarate in the tissue of fumarate hydratase knockout animals in the kidneys. This is just a representation of a kidney. And more importantly, we can find it also in the urine. So fumarate is a very important biomarker of fumarate hydratase loss and very specific. But you might be wondering, yes, why this is associated or why this drive cancer. And indeed, we now know, there is some evidence, I'm just depicting the summary here, that fumarate, when accumulated to this level, can drive cancer and can drive oncogenic signaling cascades, and I will give you a few examples, making these metabolites a bona fide oncometabolites. So this is an important concept. Metabolites are not inert. Metabolites can play biological functions. One of the most interesting for us function is related to the chemistry of fumarate. Fumarate is an alpha-beta unsaturated dicarboxylic acid, and it's prone to nucleophilic attack by cysteine residues of proteins, leading to this post-translational modification called succination. And succination is a very important feature of fumarate hydratase deficient cancer. And just to show you a few examples, we generated an antibody that can detect succinated proteins. And this is shown here. These are two fumarate hydratase knockout human clones that we recently generated, and two succinic cysteine, that is the name of this antibody, can clearly detect fumarate hydratase deficient cells. And more importantly, if you take a slice of the kidney of fumarate hydratase deficient cells and you stain the two succinic cysteine antibody in magenta, you can clearly see how it stains beautifully fumarate hydratase deficient clones. And not only that, this fumarate dependent metabolites can be seen also in the urine of the animals as 2-succinylcysteine, a breakdown product of it, and can also be seen in cancer patients. We have 2 HLICC patients. This is a very rare disease, so the number of patients is very low, but we were happy to confirm that both fumarate and some fumarate dependent markers were high in these patients compared to the normal counterparts. But why this is important? Well, one target of fumarate is this protein called KEAP1, which you might have heard of because it's the negative regulator of NRF2. NRF2 is an important antioxidant response gene, and when fumarate binds to KEAP1, its negative regulator, by succination, NRF2 can activate its transcription and reprogramming, leading to a very important antioxidant response, in the sense which is thought to be a driver of cancer in this tumor. And I'm just showing that in the fumarate hydratase deficient clones that we have generated, there is a very striking signature of 2 important NRF2 target genes called NQO1 and hemoxygenase. Now, I just wanted to give you the last, I have still a couple of minutes. 2 minutes? Yes, very quickly. 2 functions of fumarate that you might not have predicted based on this chemical structure, but more related to its biological function as inhibitor of a family of enzymes called alpha-ketoglutarate dependent dioxygenases. And then giving you this example of prolyl hydroxylases, these are enzymes that oxidize probably residues of proteins, and one of the most characterized protein that is subject to this activity is the epoxy-inducible factor HIF. So when hydroxylated HIF is degraded and therefore is not active, but when fumarate is high, this reaction, which is alpha-ketoglutarate dependent, is inhibited by product inhibition due to fumarate accumulation, and therefore HIF is allowed now to be expressed, stabilized, and we can see a very striking accumulation of the pseudo-hypoxic signature in fumarate hydratase deficient cells. I'm calling it pseudo-hypoxic because these cells become hypoxic, they have a hypoxic response, even when oxygen is present. And this is a very important feature. Another target of this activity of fumarate is another class of alpha-ketoglutarate dependent deoxygenases called TETs, which are implicated in DNA methylation. And when fumarate is accumulated, DNA demethylation is blocked, leading to epigenetic inhibition of some target genes. Here I'm just showing one recent finding that we made, whereby a family of anti-metastatic microRNA can be inhibited by fumarate accumulation, leading to an epithelial to mesenchymal transition. So, I'm going to go to the last slide where I'm trying to wrap up what I just showed you. And I think it's very important to realize that our findings where we have a mutation of a mitochondrial enzyme that causes cancer indicate that metabolic dysregulation plays a key important role in initiation and transformation of cancer. And in this particular case, fumarate, just a small molecule metabolite, can activate all this series of transcriptional and epigenetic reprogramming that can drive tumorigenesis and promote it to metastasis as well. Okay, I think here I will leave this conclusion, where I summarize basically what I just said, and to underline the importance of mitochondrial dysfunction in tumor initiation and progression. I will leave this with you so we can discuss if you have any other questions. And of course, I thank you all for listening to me.
Thank you, Christian. We still have roughly 5 minutes for a couple of questions. Anil?
Anil Modak from the US. Great talk, Christian. It appears that you conclude that mitochondrial dysfunction causes metastasis in patients with cancer.
In specific...
or results in metastasis.
In specific circumstances.
Yeah. Have you ever thought of evaluating mitochondrial function using a breath test, like methionine breath test, in patients, and then find out if there is metabolic dysfunction? Those are the patients that might progress to metastasis.
Oh yeah, this is definitely an excellent question, something that we have discussed.
It's a very simple breath test with 40 minutes and can evaluate mitochondrial function.
So my approach, which is reductionistic compared to others, would be to try to investigate whether some of these metabolic changes that we see is translating into the volatile metabolites. So we know that we have specific markers of mitochondrial dysfunction that we can pick up in a breath test. For me, that would be the limiting step, and my knowledge is that there are no such biomarkers for mitochondrial dysfunction yet, at least, but this is the perfect audience.
That's the reason why you use label methionine,
yes,
which regulates mitochondrial function. And if you see high levels of 13CO2,
CO2 as a readout.
And if it is low,
okay, that's
a patient might progress to metastasis.
This is an excellent point. Yes, yeah.
Thank you very much. It's very, very interesting. So Chris Breitling from Leicester. So mitochondrial dysfunction is also important in other chronic diseases as well. And have you sort of looked at the role of other pathways, so the role of SOT2 in mitochondria or NADPH oxidases, such as what's thought?
No, we haven't looked at specifically these targets, but actually there is another part of the animation that I'd left out that is more related to what you say. I think mitochondrial dysfunction, as you correctly pointed out, is the readout of many pathological conditions. So, for instance, we know that inflammation in macrophages when you have this change in the phenotype of macrophages, can dysregulate mitochondrial function quite extensively. And one metabolite that is strikingly accumulated when you have this type of polarization in macrophages is succinate and itaconate. So I don't know if you've heard about these two metabolites, but they accumulate to millimolar level in the cell, and they have important antimicrobic functions, and they can be used by macrophages to kill when bacteria are engulfed. So these are important readouts of mitochondrial function in macrophages, for instance. And there is plenty of evidence now that this can be used as markers. We use them as markers in many diseases. We found it in multiple sclerosis. We found it in classic sepsis. Yeah. I hope I addressed your question, or was it?
Not entirely, but it was still interesting.
Okay, yeah. Well, we might discuss it.
Can you comment further what's driving the selection of cells that have a poor metabolic rate? Are they escaping normal oxidative stress? That is a way to kill bad cells in our body? Is that the driving for selection of these cells?
I wish I had the answer. I think this is one of the billion-dollar questions that is in the field now. Because, you know, there might be many triggers. One trigger is the hypoxia. So when the tumor grows, it becomes hypoxic. So it is possible that if you have a mitochondrial dysfunction, therefore you don't need oxygen for fueling the cell, and the cells are already pre-adapted to that hypoxia, then the cancer cell survives better. The other hypothesis that is around is that the tumor survives better with this type of mitochondrial dysfunction when it's in nutrient-deprived condition. So it's also a matter of nutrients and a matter of oxygen that might drive this evolution, but there is no clear answer to that.
Thank you, Christian.